Welcome back to the CD Genomics Next Generation Sequencing video series. In this video, you will learn about the most widely used methods of DNA methylation sequencing and their comparison. DNA methylation is an important component, as well as a research hotspot of epigenetics. The dynamic DNA methylation demethylation process plays critical roles in gene expression control and cell development. DNA methylation is one of the first identified forms of epigenetic modification of genes and may present in all higher organisms. DNA methylation can turn off the activity of certain genes, and demethylation induces reactivation and expression of genes. The major forms of methylation are 5-methylcytosine, N6-methyladenine, and 7-methylguanine. In eukaryotes, the DNA methylation only occurs on the cytosine residuals and specifically for the CpG sites. DNA methylation and hydroxymethylation at the 5th position of cytosine are essential epigenetic alterations that play vital roles in gene expression regulation and cell differentiation. DNA methylation typically represses gene transcription. Hydroxymethylation, on the contrary, activates gene expression or promotes DNA demethylation. Cytosine DNA methylation and 5-hydroxymethylcytosine genome-wide mapping uncover the genomic areas of these alterations, which is crucial for their mechanisms to be elucidated. Cytosine DNA methylation groups introduced by DNA methyltransferases can be iteratively oxidized to 5-hydroxymethylcytosine, 5-formylcytosine, and 5-carboxylcytosine by the action of TAT-methylcytosine deoxygenases. 5-hydroxymethylcytosine is the most abundant component in vivo among the 3 5-methylcytosine oxidative derivatives, and can be identified in almost all mammalian tissues and cells. 5-hydroxymethylcytosine is now regarded as an epigenetic alteration in relation to being one of the medium states of DNA demethylation. The oxidation derivatives of 5-methylcytosine could impact biological functions by altering DNA properties or recognition by potential reader proteins. This dynamic process is critical for cell reprogramming and development. Aberrant methylation is a common feature of many human disorders, including cancer, autoimmune diseases, heart diseases, skin diseases, etc. Therefore, this dynamic process has been considered as an important factor for studies of tumorigenesis mechanisms and the discovery of therapeutic targets. With the development of next-generation sequencing technology, we can analyze events such as 5-methylcytosine to 5-hydroxymethylcytosine and other modifications at a genome-wide level. This is called the DNA methylation sequencing, which allows us to get access to even more information that traditional genomics research cannot. DNA methylation sequencing methods can be divided into 3 main categories according to their principles. 1. Bisulfite sequencing. 2. Restriction endonuclease-based sequencing. And 3. Targeted enrichment methylation sequencing. A variety of different sequencing methods have been derived based on the previous principles. Some representative methods include bisulfite sequencing, reduced representation bisulfite sequencing, oxidative bisulfite sequencing, TAT-assisted bisulfite sequencing, and methylated DNA immunoprecipitation sequencing. More sequencing methods are also listed. Bisulfite sequencing, oxidative bisulfite sequencing, and TAT-assisted bisulfite sequencing allow for the methylation analysis at a single base level. Depending on the sample processing method, these 3 sequencing methods can detect 5-methylcytosine and/or 5-hydroxymethylcytosine. Bisulfite sequencing uses bisulfite to treat DNA to deaminate unmethylated cytosine into uracil, which is then sequenced. The bisulfite-treated and untreated sequenced samples are compared to find the methylated sites. However, bisulfite sequencing is unable to distinguish between 5-methylcytosine and 5-hydroxymethylcytosine. In the oxidative bisulfite sequencing process, 5-methylcytosine was retained, while 5-hydroxymethylcytosine was oxidized and then deaminated to uracil for sequencing. By comparing the sequencing of oxidized and untreated samples, 5-methylcytosine and 5-hydroxymethylcytosine can be distinguished from each other at a single base level. TET-assisted bisulfite sequencing uses beta-glucosyltransferase to protect against oxidation of TET protein by 5-hydroxymethylcytosine. 5-methylcytosine and unmethylated cytosine are deaminated to uracil, which allows the identification of 5-hydroxymethylcytosine. Thanks for watching this video. If you like more information on sequencing methodologies, please visit our website, www.cd-genomics.com. We look forward to assisting you.